So as we come full circle in this chapter, I wanted to kind of summarize some of the features that we are going to see on the homework and the exams. This is also kind of represented in Supplemental 10 in the back of your lab book. So as we look at proteins, let's start to think about what's most important about proteins. So when we look at proteins, let's first look at monomers. So when we think of monomers for a protein, we need to think about amino acids. And amino acids, there are 20 of those. And of those 20, 10 are essential. So 10 you have to get from your diet. Talked about that. Next thing we want to know is the functional groups. And it kind of gives it away in the name. You can imagine that from an amino group is the amine, which is the RNH2 group. And from the acid, we can get the carboxylic acid group. That's R-carbonyl OH. Bring those together and kick out water. So losing water makes this a condensation polymer. Gives us our linkage. And a linkage, in this case, would be the RNH group. Lose water, you get carbonyl, and then the other R group, and that's the amide. What's most important, though, is amides, even if they're polyamides, in biochemistry, they are called peptides. And that's what we need to know for the homework and the exam. So know the monomers, know the functional groups, know the linkage, and that's guaranteed, 3 or more questions. It's also important to know that proteins are where we get our enzymes, and those typically end in ASE. So watch out for those. It's also important to know that proteins start with P, and so do peptides start with P. So you'll see that kind of relationship used in this particular chapter. So as we look at proteins, again, those are amino acid based. Let's look at carbohydrates next. Carbohydrates based on sugars. So when we look at carbohydrates, remember that means carbo is carbon, hydrate is water. So here's carbon, here's water, that's carbohydrate. When it looks at monomers, we need to know that the monomer most often is glucose, and that's a monosaccharide. It's also true that other things like fructose and galactose would fit in that category too, just not for purposes of our introchem class. Glucose is the most common. When it comes to functional groups, what we're looking for is all sugars have aldehydes and ketones. Aldehyde is R-carbonyl H, has H in the name. And ketones have an R-carbonyl R, so different being edge versus middle for that carbonyl position. They also have alcohols. So notice these groups are very different than amino acids in the previous section because they, these groups are very different. There's no nitrogens, for example. So whether you take an aldehyde or a ketone, it's one, not both, one of those, and lots of alcohols give us our linkage. And the linkage here is what we call an ether. It's an sugar on ether side, so to speak. But biology calls it by a different name. They call it a glycoside, and that's the one that we need to know and recognize in this chapter. We also said that carbohydrates are just another name for sugar. And sugar and saccharide start with S. So it's easy to remember those. It's also important to know that sugars end in OSE, not ASE, like amino-based protein enzymes. So very different groups, very different monomers, very different linkages to know for carbohydrates versus proteins. Let's look at our third category, lipids. So lipids are not polymers. They're not going to have a monomer. They're going to make up our fats, waxes, and oils. So let's look at those in the same kind of capacity. So for a lipid, when we say monomer, that's a trick question because they are not a polymer. As we go to the functional groups, though, they do have groups that relate back to organic chemistry, and that's why we cover this chapter so close. Functional groups are going to be alcohols. Alcohols, in this case, are going to be ROH, which is a disadvantage because we also see alcohols in carbohydrates previously, so you got to be careful with this one. And we do have some carboxylic acids, which is also painful because they too show up in proteins, but with amines, not with alcohols. So R-carbonyl OH. Put those two together, and you get an ester. An R-carbonyl OR. And that's the linkage. And luckily, they don't change it. They call it an ester before and after biochemistry versus organic chemistry, which is nice. Lipids are nonpolar. It's the only group of the bunch that's nonpolar. Everybody else very polar. And also we have a variety of different types. We have fats, oils, and waxes. In a fat, you're typically from an animal. You're in that triglyceride kind of format, but you're also saturated in your hydrocarbons on the fatty acid. An oil is typically from a plant, and it's typically unsaturated. And still has that same triglyceride kind of format of glycerol and 3 fatty acids. A wax, however, is no glycerol. It has a long chain alcohol instead. So no glycerol there. And it tends to be saturated, which is the worst of both worlds. And that's what we need to know about lipids, not polymers. Functional groups, unfortunately, do have some overlap with both carbohydrates and proteins, but they make esters in the end. Our last major category is the nucleic acids. Nucleic acids is what makes up all living things, gives it their genetic code in order to build more of those living things. So as we look at nucleic acids, remember things like DNA versus RNA. When it comes to monomers, it's pretty complicated. We have sugars, which is going to be an overlap with carbohydrates because carbohydrates are sugars. We have amine bases, which will be overlapped with proteins because those are amine base and carboxylic acid based. And then we have the phosphates, which is an T4-3 ion from exam 2. So when we look at functional groups, functional groups are based on those organic groups that we saw in a previous chapter. Sugars could kind of tell us that we're going to see alcohols. That's what's going to make them so polar. We did mention that sugars have aldehydes and ketones, and that's true in their kind of chain form, but all sugars are in their ring form in DNA, so we're not going to see those aldehydes and ketones present, so we can ignore those. The amine bases, just given the name, you can recognize the amine group, but they also have amide groups. Remember D for double bond. The amine itself is the RNH2 group, and the amide is the R carbonyl double bond NH2 group. And then the phosphate is actually the linkage that holds those sugars together. So phosphate actually comes down here. That's that PO4 -3 kind of group, phosphate. It's in the 4 box, it's got 4 oxygens. When it comes to the amines and amides, just remember you have the A's and T's that have 2 hydrogen bonding sites, the C's and the G's that have 3 hydrogen bonding sites, and that's for DNA. Thymine, the T, replaces with U, uracil, and RNA, but it still has 2 hydrogen bonding sites. All very polar, all recognizable as the genetic code in this twisted ladder we call DNA. So you got to be a little bit careful here because there is some overlap with all the groups. Only amino acids are the ones that do not have alcohol, so all 3 other groups do, so be careful when you remember that one. And then amines and amides definitely show up in the amino acids of proteins, but now the nucleic acids with DNA as well. So if you know the monomers that repeat themselves, if you know the functional groups and the linkages, that's guaranteed to be about half the information you need to know from this chapter moving forward as we get to exam 3 coming up. So our chapter next time will be over the glue that holds all these molecules together. We talked about it already, called hydrogen bonding. That's the major focus of that chapter. But there's also dipole forces and generic dispersion forces that we'll talk about too. And we'll catch you then.